Professor Ariel First in the Chemical Engineering Department, and it is my honor today to introduce Professor Shiny Vargas, who is a professor of biomedical engineering, mechanical engineering, and materials science, as well as orthopedic surgery at Duke University. She's also the inaugural MEDEX investigator at Duke. In 2016, she was inducted into the College of Fellows for the AIMBE, and she's an associate editor of the Journal of Biomaterial Science. And there's a whole list of awards and honors that I'm not going to go through today, but I'm very excited to hear about her research, which includes biomaterials and bio-inspired systems, as well as regenerative medicine. So Shiny, if you want to get started, thank you so much for speaking today.
Thank you so much for the invitation and Ariel for that wonderful introduction. Today, as Ariel mentioned, I'm going to talk about our research in the space of biomaterials. Before going into the talk, I'm going to just introduce various research that we have in our lab. A major area is designing biomaterials. There, the idea is to control interaction of the cells with their extracellular matrix and how we can use the physical and chemical properties of the ECM to drive self-renewal versus differentiation. So you can see that the system here, this is a lung organoid where we are looking at how we can culture the lung precursor cells, that is AT2 cells, or use biomaterials to differentiate them into AT1 cells and create a lung organoid. And here it is a skeletal muscle, and here it is differentiation of stem cells into osteoblasts. Now, you know that in the case of diseases like a tumor, cancer, fibrosis, etc., or even the scarring, the physical and chemical properties of the ECM changes. It could be the increasing the mechanical properties, ECM chemical composition. So in those cases, the interaction of the cells with their extracellular matrix is perturbed. So we are also interested in understanding how the cells respond to the signals that contribute to disease progression. Moving forward, we like to use these understandings to create new therapies. In fact, one of the approaches can be use the ECM as a signal to resolve fibrosis. In fact, we have done some work in skin fibrosis. And today I will be talking about how we developing therapies for promoting fracture healing or treating osteoporosis, that is a bone degeneration diseases. We also have some work in the space of cell transplantation. So how we can use biomaterial as a device and to improve the host tissue interactions and also keep large number of cells. And also creating model systems, mainly with organ-on-a-chip technologies, which I will not be talking today. Today in my talk, I'll be focusing mainly using biomaterials as an artificial ECM. So we create biomaterials with a defined physical and chemical cues to recapitulate tissue-specific milieu. In this tissue-specific milieu is used towards organoids, cell transplantation, and also for organ-on-a-chip systems. Here we are showing an example where we use the biomaterial to support the self-renewal of ESCs and iPSCs. And in this case, we use the biomaterials to drive osteogenic differentiation of stem cells. Here it is the skeletal muscle, and here it is the lung tissue. So here we are looking at a precursor from AT2 and creating a lung organoids, and this is unpublished. So as I said, today in my talk, I'll be mainly focusing on generation of biomimetic materials as a tool to study the molecular mechanism and identify new therapeutic targets, and finally, how we can use this knowledge to support in situ tissue repair. Here today, I will be focusing on mimicking bone extracellular matrix. So as I said, we will be focusing heavily on the ECM, and in this case, we will be mainly focusing on calcium phosphate mineral environment. As you all know, the bone is a heavily dynamic tissue. There is excessive remodeling. Whenever the two cell populations are working in concert, the osteoblasts contribute to the tissue formation, osteoclasts, promote ECM degradation. So the ECM degradation also contribute to the dissociation of the calcium phosphate into calcium and phosphate. And when you have a lot of calcium and phosphate in the extracellular milieu, they can precipitate back and form calcium phosphate. So we wanted to create this dynamic environment. So we use the biomineralization approaches to create a scaffold or a biomaterial with the calcium phosphate minerals. And in this case, we created a hydrogel. This is more like the soft contact lens that you use. And everyone knows a lot about hydrogel, so I'm not going to go in detail. We created this hydrogels with this dangling side chains. So this dangling side chains has a COH group, that is a carboxyl group. So when you expose them into calcium and phosphate, this CO group can bind to calcium and promote calcium nucleation. And you can see that changes the color. So we know that these materials are mineralized. Then we can characterize these materials, etc. Then we used this material to study the effect of these materials on promoting osteogenic differentiation. Here we have used MSCs. These are from bone marrow derived MSCs, embryonic stem cells, and induced pluripotent stem cells. We have covered their we compared their differentiation into cover slip and non-mineralized material as a control. And you can see when you culture these cells on mineralized material, that is the green, you see an upregulation of osteogenic markers, and all these cultures were done in growth medium. So we don't use any osteogenic inducing soluble factors. We just wanted to look at the osteoinductivity of this material. So we did an ectopic bond formation. So we implanted this scaffold into the subcutaneous space. So here is a non-mineralized, no cells, mineralized, no cells, and mineralized with HMSCs. And we looked at them after 4 and 8 weeks. And you can see that you don't see much when there is no minerals, no cells. However, in this case, you can see mineralized, no cells, mineralized with HMSCs. We see this vascularized tissue. And if you look at the histology, they look more like bone tissue, and they are also positive for osteocalcin. That is a specific marker for osteoblasts. We also looked at the ability of this fusion. So this is a spine fusion. So we did a surgery and created L4, L5 transverse process and used the biomaterial to fuse the spine. And this is where we have done 4 weeks saline. So we take the graft, soak them in saline and implant them. This is graft with marrow. We also have growth factors BFGF and BMB2. And we saw bone formation in all grafts, irrespective of whether we had the growth factors or bone marrow derived cells. In fact, we did not see any difference between biologics. We saw whether we had marrow, growth factors, or just the saline. All of this graft promoted bone formation. This means the synthetic biomineralized material recruited host cells and contributed to bone tissue formation. And in all these cases, we see a fusion, but that is not as good as autograft. Autograft was always better. We also asked a question, can we use this approach to support bone marrow transplantation? So here the idea was that can we use this as a scaffold to transplant bone marrow in the ectopic bone space? So we created a dual compartment structure. So you have an outer layer that has a mineralized layer, and then you have an inside that is non-mineralized, and we load the bone marrow cells. So the idea is that, okay, can we just transplant them into an abdomen or somewhere is an ectopic space, right? So the outer layer, that is the mineralized layer, we leave it acellular. That means this upon implantation, this can recruit cells from the host and form a bone tissue. So this is before implantation. You can see we create something like a donut. So outer layer is mineralized, inside is non-mineralized, and this signal is not sufficient to pick up in microCT. This is post-implantation. After 6 months, you can see you still have the donut. The outer layer is much more dense, so we can pick them in 3D microCT. So that means there is a cell recruitment. Here we look at the histology. You can see outer layer forming bone-like tissue, inner layer is fat, and we also see the bone marrow containing long-term HSCs and short-term HSCs. So this approach allows us to support the bone marrow transplantation in vivo. Now, given that the material is very osteogenic and osteoinductive, we wanted to look at the molecular mechanism, started looking at the molecular mechanism. So we have a calcium phosphate minerals. We engineered this material to dissociate into calcium and phosphate. So now we have a concentration of calcium and phosphate that the cells are exposed. So we wanted to look at the mechanism by which this drive osteogenic differentiation. So there are enough studies looking at calcium ions. So we focused on phosphate ions. So our focus was on SLC20A1. That is a phosphate transporter. So we wanted to look at whether we can control the transporter and thereby control the uptake of phosphate into the cells. Now, first we looked at the expression level of SLC20A1, that is a phosphate transporter, on a mineralized. So this is cells on mineralized. This is cells on non-mineralized. And you can see higher level of SLC20A1 in a mineralized material. Western blot to look at the expressing SLC20A1 compared to the non-mineralized material. Now, this gives us a knob. Now we can use this knob to control the phosphate intake and thereby study the molecular mechanism. So we have used siRNA. So we have scrambled siRNA, and this is SLC20 knockdown. And we first looked at the knockdown efficiency. So first we looked at regulation of SLC20A1, and both that is although in the case of non-mineralized, and concomitant with this down regulation, we also see a down regulation of osteopondin and osteocalcin. And we also looked at the protein level, and you can see this is the mineralized material, and this is you see more osteocalcin on mineralized material, scrambled siRNA, you don't see any effect, and you see the downregulation of osteocalcin in the case of SLC20A1 siRNA. So this means mineralized material assisted osteogenesis was diminished upon SLC20A1 knockdown. Then we looked at, now we have in controlling the phosphate uptake. So we wanted to see it. So we looked at intracellular phosphate on mineralized material. With SLC20A1, we see a down regulation. The same with the intramitochondrial. We also see a similar effect with intramitochondrial phosphate. That means an upregulation in the case of mineralized material, and the level went down with siRNA treatment. Since phosphate is a precursor for ATP, we also looked at ATP level. Again, you see the same trend. We have also stained them from quanacrine staining, that is looking at intravesicular ATP. And we see the same trend there too. So whenever you have higher level of intracellular ATP, it gets secreted into the extracellular space, and now it acts as an extracellular ATP as a signaling molecule. So we wanted to look at how that is controlling the osteogenic differentiation. So we used N-ethyl-maleimide, that is an inhibitor that can be used to inhibit the vesicle transport. So you can see that when we use the NEM, that is, we see a downregulation of osteocalcin, osteopondin, and we see the same in the case of the protein. This means the intracellular phosphate has to be converted into extracellular phosphate to have an effect, right? So we started looking at extracellular ATP as a signaling molecule. We looked at purinergic receptor because that is a receptor which extracellular ATP uses. So we used pharmacologic inhibition of purinergic receptor by suramin. And so when you have no suramin or plus suramin, we see no effect. We see osteogenic differentiation in both the cases, whether we block the purinergic receptor or we don't block the purinergic receptor. So we went and looked at the level of ATP in the medium, but we could not detect anything. So there was not very detectable levels of ATP in the extracellular medium. So then we wanted to go back and check what is happening. So you have intracellular ATP. The intracellular ATP cells are also positive for CD39 and CD73. So they can metabolize the ATP into adenosine. So we started looking at the presence of adenosine. And so when we look at the adenosine, you can see there are higher levels of adenosine in the medium when we culture the cells in mineralized material. And when we use the siRNA knockdown, you see a downregulation. So we know that at least the ATP is getting metabolized into adenosine. So we next looked at the role of adenosine as a signaling molecule. So in fact, we have looked at two receptors. One is A1 receptor and the other one is A2B receptor. When we use the A1 receptor, we don't see any effect. When we target the A2B receptor, we see a downregulation. If this is true, then we should be able to take a non-mineralized matrix, so that does not promote osteogenic differentiation, add adenosine into the medium, and that should give us an osteogenic differentiation. So we started looking at whether we can use adenosine, exogenous adenosine, to drive osteogenic differentiation. And you can look at non-mineralized material, not much osteocalcin expression. We add adenosine, the expression level goes down. We perturb the A1 receptor, no effect. But when we block the ATP receptor, we see a downregulation. We see the same with osteopondin. We see the same in the case of osteocalcin protein level. So whenever we block the ATP receptor, we see the downregulation of osteogenic differentiation. This tells us that pharmacologic inhibition of ATP receptor downregulate both the mineralized material and exogenous adenosine-mediated osteogenic differentiation. Then we asked the question is that can we use exogenous adenosine as a small molecule to promote differentiation of stem cells? So for that reason, we have used ESCs and iPSCs, cultured them in growth medium. You don't see any differentiation. Now you just add adenosine into the medium, you see they undergo osteogenic differentiation. So by just using the adenosine as a small molecule, we were able to derive osteoblasts from ESCs and iPSCs. This allowed us to propose a new molecular mechanism. You have biomineralized material, releasing calcium and contribute to ATP and finally metabolized into adenosine. Now the adenosine act as a paracrine or autocrine molecule through A2B receptor, supporting osteogenic differentiation. Now, after that study, this is not from our group. This came out where they looked at the effect of A2B receptor in a mice model. So this is an A2B receptor knockout mice model. And they noticed that the bone density is low when the A2B receptor is knocked out in this mice. And if you take the bone marrow progenitor cells from this mice and try to differentiate them into osteoblasts, they undergo less differentiation compared to control. So we were very happy that the biomaterial mediated finding can be also mimicked in the animal model. We next ask the question is that mesenchymal progenitor cells can also differentiate into fat. So what is the role of A2B receptor in fat formation? So what we did is that we took mineralized material, cultured bone marrow derived MSCs on the material and introduced them into adipogenic inducing media. So now we have a non-mineralized material, we have a mineralized material, we have cover slip, and we culture them in adipogenic inducing medium. And you can see that the cells on non-mineralized material and the cells on the cover slip show positive for fat formation, that means genic differentiation. Now on the cells on the mineralized material does not show any positive staining for perlipid. So in fact, they are instead, when we stain them for osteocalcin, that is a marker for osteoblasts, we do see that the cells on the mineralized material are positive for osteoblasts. That means the cells, calcin, that means they are, when we block the A2B pathway, the cells on the mineralized material, so these cells can become fat when we block the A2B pathway. So when the A2B activated, the cells go to osteoblasts, or osteogenesis, when you block A2B, they go to fat. So that was something interesting to us. Now, we also asked a question. Now we know that adenosine plays a role in osteogenic differentiation and building more bone. So can we harness, as you all know, adenosine is a ubiquitous molecule. That means A1 receptors are present throughout the body or targeted therapeutic. First idea is that can we leverage endogenous adenosine, because we know that the cells secrete adenosine. So can we just leverage this endogenous adenosine? However, that is tricky because adenosine in the tissue is very, very low levels. And also this adenosine is very transient. In fact, if you look at the circulation, the half-life is less than 10 seconds. We know that whenever you have a trauma or there is an injury is introduced, you see a surge in extracellular adenosine because the cells under stress secrete a lot of adenosine. So our next approach was that, okay, can we look at with the fracture, there is higher levels of adenosine in the bone marrow. So this is the pre-fracture. Now we introduce a fracture, there is an adenosine surge. So you can look at, this is the pre-fracture level, and this is, we measure the adenosine as a function of time, and you can see that higher level of adenosine with post-fracture is almost 10-fold. So the initial 1 to 2 days, we see almost, or like roughly in 1 day, we see almost 10-fold increase in extracellular adenosine following injury. So now we wanted to use this adenosine surge or sequester this adenosine so that we can localize them at the fracture site and use it to promote the fracture healing. So we created an injectable biomaterial or a patch. So initial one was a patch. So you have a broken bone. So we have a patch that can sequester the adenosine, keep the adenosine at the fracture site, and see whether we can promote the fracture healing, right? So we have done, so what we did is that we used polyethylene glycol because it's bio-inert. We modified the polyethylene glycol with boronic acid. The boronic acid can form cis-diol dynamic bonds with adenosine so that we can complex them, one way to sequester them or load them and then release them. So that is what we have done. And so we have, here is a PEG. So we don't see much adenosine sequestered after 3 days. This is a PEG modified with boronic acid. We see higher level of extracellular adenosine following 3 days. So in fact, we are able to sequester them and localize them at the fracture site. In fact, we looked at how the effect of the sequestration of adenosine on fracture healing. So this is PEG, no boronic acid. So that means there is no significant sequestration. This is 1 molar boronic acid. So that means they can sequester what is there in the tissue. And we also have one group where we load them, preload, and so we looked at the fracture. So this is where the fracture, so we can see the fracture site, so we can follow them, X-ray and micro CT. This is after 14 days. This is after 21 days. And after 21 days, we took the fracture section and looked at the images and quantified them. And you can see that we see a better bone building or bone formation as we increase the adenosine content. So this group has a higher adenosine level compared to this group because this is only sequestration. This group contains sequestration plus loading. We also looked at the vascularization, and in fact, this is endomucin staining. And you can look at, we did not see any dose-dependent effect. Instead, we saw that the adenosine, wherever we sequestered the adenosine or locked, we saw higher vascularization at 7-day and 14-day compared to just the PEG. And 21-day, there was no effect. So the effect on vascularization in the early days, like 7 days and 14 days. We know that with aging, the healing ability of the tissue decline. So we wanted to look at what is the effect of, is there a role the extracellular adenosine play with aging? So we looked at the young and old animal, both male and female. You can see that there are less adenosine in the old cohorts compared to the young ones. Then we took the bone marrow derived MSCs from these or mesenchymal progenitor cells from these mice, both young and old. This is in growth medium, so there is no significant osteogenic differentiation. We cultured them in adenosine, and you can see that all of them under osteogenic differentiation of old cells or cells derived from the old mice. So now towards this, we wanted to now use adenosine to promote healing with aging. So instead of creating a patch, we created these microcarriers or microgels loaded with adenosine, and they also have functional groups. So when after injection, they cure and form a scaffold. And in fact, our initial studies, this is the control. Without adenosine, this is adenosine treated. They undergo secondary ossification, so that is endochondral ossification. So you can see that there is some cartilage here, and adenosine is pretty much healed. So you can see the fracture here. This is pretty much healed, and this is just the quantification. We also looked at the vascularization, and this is the adenosine treated group. This is the control, and you can see more vascularization. This is after 21 days, and this is after 7 days. We also noticed that this therapeutic formulation also have a role in mitigating fracture pain. So in fact, this is the adenosine treated group working this location therapeutic formulation. From this, we can calculate something called duty factor. So this is the ipsilateral, that is the fractured leg compared to the contralateral unfractured. And this is the control. You can see that they have less movement when you introduce the fracture. Now this is the adenosine treated group. You can see they have comparable movement. So they can use the leg more. And in this case, we are comparing the control group that is non-fractured, and this is the adenosine, control group fractured, but no adenosine, and this is the fractured with adenosine. And you can see that they can move the leg more. Here looking at the movement time, and they have more functional when you treat, the animals are more functional and they move more when you treat with this therapeutic formulation. We also looked at their weight bearing ability, whether they can stand on their fractured leg. And here you can see that when you treat with adenosine, they have more weight bearing ability compared to the control, that is the non-adenosine group, treated group. So it looks like adenosine and this therapeutic formulation really mitigate early fracture pain. So we only do fracture pain up to 10 days. We also isolate the DRG from these fractures, one with the adenosine treated and one with the control treated. And then we introduce them to capsaicin and look at their ability to tolerate the pain. And it looks like with adenosine treatment, they have less activation of membrane potential compared to the control. So this is the in vitro study validating what we saw in the animal study. We also looked at the role of extracellular adenosine in bone health by looking at osteoporosis. And this is overactomized mice. And we see decreased extracellular adenosine in obese mice. And this is driven by estrogen. And as you all know, osteoporosis is characterized by low bone mass. And you see an imbalance between bone formation and a higher activity of osteoclasts. So you have a lot of bone resorption and less bone formation. So the most of the current drugs is bisphosphonate-based drugs. They target the bone resorption. That means they target the osteoclast activity. There is no bone formation. There is only bone preventing further bone resorption. So we wanted to look at the effect. We know that extracellular adenosine can promote bone formation. We also wanted to see that the role of adenosine on osteoclasts that contributes to the bone resorption. And we know that we found that extracellular adenosine, in fact, down-regulate osteoclast activity. So this is a number of markers we are looking at for osteoclast activity. And here looking at A2BR, the role of A2BR signaling. And we notice that this down-regulate osteoclast activity. So we asked, can we use adenosine-based drug as a new drug? Can we have a new drug? And in this case, it promotes bone formation because it's osteoanabolic. And it also has a role in down-regulating osteoclast activity. So it will also prevent bone resorption. So we will have a balancing act. So in fact, in this case, we have to now think about how to deliver adenosine systemically. So we have created a nano carrier. This is a hyaluronic acid-based nano carrier. We have a bone binding unit, that is the alendronate, which will bind to the bone. And then we have a drug loading. This is the PBA or the boronic acid. So we can load the adenosine into the nano carrier and deliver. And we have done an animal studies. We have a control that is a healthy group. Then we have OVX mice, that is the one which display osteoporosis. This is a group where they have osteoporosis and we treat them with nano carrier, and then we have a nano carrier with adenosine. And this is a number of parameters that we measure to figure out whether there is a bone degeneration or bone building. And I'm just going to focus on control and OHA because control is the healthy one. And you can see that when you measure the bone mineral density, the group that treated with adenosine is very similar to the control. The same with the bone volume. We see the same with the trabecular number because it's the vertebrae. We also see the same with the trabecular thickness, the connective density, trabecular space, etc. So it means that when we treat them with adenosine, they not only stop the bone resorption, but helps bone building. That means it's analysis which shows new bone formation when we treat it with adenosine. Now, the rest of the time, I am going to really tell you guys about some can make this hydrogels self-healing to these biomaterials. So we can, most of these hydrogels that we use, so they don't heal themselves. That's because they have irreversible crosslinks. And now, since they have irreversible crosslinks, they lack mobility. The chains don't move. So they cannot reptate, or they cannot go and fuse each other, the polymer chains, so they cannot entangle. However, a number of studies have given self-healing functions to such networks by chemical reactions, secondary interactions, multi-component system, etc. However, most of the hydrogels are in aqueous system, so it is very difficult to have these reactions incorporated into the hydrogel system. So we asked the question is that, okay, we know that this is the chemically crossed in hydrogels. So you are looking at, this is the polymer backbone, that is the red, and this blue is the cross-linker. So the two of these polymer chains are connected by this cross-linker, so they don't move. But what if I can provide a dangling side chain? So I have a side chain, so that can move. So here the idea is that when the backbone doesn't move, they stay, but this side chain is going to move. So can we use this side chain movement? So I have one hydrogel with this side chain and another hydrogel with this side chain. So when they come in contact with each other, can they stick to each other and heal? So that was the idea. And in fact, as you know that here we use COH group. So if you look at the end of this side chain is carboxyl group. So we can also use pH as a trigger to control their healing, which I will explain later. So when at low pH, so this is the two hydrogel piece. We just colored them just for the better visibility. At low pH, you can see they stick to each other. Now we can take multiple pieces. So that means here we have multiple pieces, they are sticking to each other. That means they can carry their own weight and the interface is pretty strong. And in this case, we have a 3-piece hydrogel, we are stretching them. And you can see that the middle portion is getting stretched, but it is not breaking. And in fact, if you look at it, always break where we put the finger because of the stress. So, and this is reversible. When we increase the pH, it goes back to its unhealed or unsticky stage. So this is a movie video which shows the healing. And this is where we create the hydrogel. We move it to a acidic temperature. So it's pH 3 here. In fact, this should happen below pH 4.5 because the pK of the carboxyl group is 4.6. So you can see that repeated stretching, the interface is strong, nothing will happen. It looks very elastic and it can tolerate the repeated stretching. So what is happening here? Above the pK or at higher pH, we have a deprotonated carboxyl group, right? CO- groups. So when you bring two hydrogel pieces together, there is an electrostatic repulsion between the deprotonated group that prevents them from coming together and healing. Now, at pH 3, we protonate this carboxyl group. Now we have a COH group. Now they can form hydrogen bonding. So when we bring them together, so we are bringing this molecule together, they come so perfectly that they can form this interleaved hydrogen bonding. So they form this interleaved hydrogen bonding, which makes this hydrogel to be stronger at the interface. Now we are using this approach a little bit to figure it out whether we can create biomaterials that can be biomaterials with improved function. So one of the biomaterial which we wanted to look at is lubricants. As you all know, hyaluronic acid is used as a lubricant to treat cartilage injuries. And this is in fact one of the HA-based lubricants where we wanted to create HA with these amino acid dangling side chains and also introduce self-healing molecules or self-sticking moieties. So you can create a shear thinning polymer that can heal and reorganize as needed after you inject them. So we are trying to improve the retention time. So if you look at the HA molecules, once you have a cartilage injury or an arthritis, they give a HA injection, but they disappears immediately. So we are trying to improve the retention time of these lubricants. So now the physical interactions allow them to form networks. Now we have self-healing. So even if they break, they can come back and heal like I show in the other movie. Now they are shear thinning, so you can easily inject them. In fact, this shows that this material can be injected through a 26G needle. So it's very shear thinning. And we also look at, this is the modified HA, this is the HA, this is the saline group. We are looking at the coefficient of friction. Low coefficient of friction means enhanced lubrication. Here we are using S-plants. So we take 2 S-plants, introduce these lubricants and look at their coefficient of friction under shear. And we also labeled this material, injected them into the rat joint knee. So here we create an ACL injury into the rat joint, and then we inject these materials into the rat joint. Here we used a low molecular weight HA because we wanted to see whether we can even convert the low molecular weight HA into a high molecular weight, right? Giving them the function because of the self-healing. You can see that when this is the modified HA, that is a self-repairing or self-healing HA, and this is the unmodified HA. By day 5, most of the HA is disappeared from the joint. And even after 20 days, we see significant amount of HA present in the joint in these animals, like even despite the ACLT and the inflammation. ACLT means the ACL injury. We also looked at the contra-protective function. That is, we left this animal treated for once a month and left them for a 12 weeks, and then looked at a cartilage at the end of the 12 weeks. And this is the contralateral, that means the uninjured but treated with the self-healing HA, and this is the unmodified HA. And you can see most of the cartilage is gone from the when we use the HA, which is the unmodified HA. And in the case of an HA, when we use a self-healing HA, it is very similar to contralateral. We have a large variability among animals. In some cases, we see no, like the majority of the cartilage is intact. There is always one animal which is not very good, etc. So there is some variability among the animals, unlike the contralateral, that is the uninjured leg. In the recent days, we also started looking at some of these, using some of these stimuli responsive materials to create more or introduce more functions into soft robots. And this is a dragonfly robot that we created. This is just a shape, nothing else. So the idea was that can we create the stimuli responsive functions into this, the body of this robot or abdomen or other body parts. In this case, we have doped the wings with a temperature sensitive material. So when you change the temperature, the color of the wings changes. We also use the self-healing material that I showed you previously, not the HA one, the pH one, where the 2 hydrogels stick to each other. Because they are responsive to the pH, we created an injury into the wings of the robot. Now, when they see the low pH, these wings heal. So thereby they create a torque that make the robot to move in the other direction. So we can control the movement of the robot by just creating a pH into the water. Changing the pH of the water surface, because we are made this robot to skim over water surface. We also created super hydrophobicity and micro architectures into the abdomen and the wing, so we can it can absorb the hydrophobic impurities. In this case, we looked at the oil because we were looking at, we can introduce a colored oil and look at how that changes. And this video shows you that because we were creating a skimming robot, and you can, and here we are just changing the acidity, introduce acidity, so the robot changed its motion into the other side. So you can have an environmental control and also as a user control. With that, I would like to stop my talk by just summarizing that we have created a mineralized molecular mechanism that contribute into osteogenic differentiation, that is involving calcium phosphate, ATP, adenosine signaling axis. In fact, we identified the role of ATP receptor in promoting osteogenesis and down-regulating adipogenesis. And we also established a therapeutic formulation involving adenosine to promote degeneration. And I also showed you some pictures, at least rough data, showing how we are using self-healing, how we are creating self-healing biomaterials and their application both in the soft robot space as well as in the biomaterial space. I know I speak very fast, and in between my internet was giving me some trouble, so I spoke way faster. So my sincere apologies for that. And I would first like to thank my group. They did all the work. I took their work. I just put it on the slides and I just came and told blah, blah, blah. So all the credit goes to them. In fact, I'm very blessed with a diverse group of students. They are very driven and I'm very lucky to have them. And all the funding support and before coming to Duke, the data that I presented was also from UCSD. I would like to thank you for speaking so fast. With that, I would like to take any questions you may have.
Thank you so much, Shiny. That was very cool work. So for Q&A, feel free to either unmute yourself and ask a question or put it in the chat and we can read it out.
Hello, Professor Shiny. That was a great talk. I was just kind of curious about the mechanism of lubrication. It's just something I have no familiarity with. I understand the hydrogen bonding giving you that retention from the shear thinning, but I don't know how you guys can predict or engineer like a low coefficient of friction material.
So we have a HA backbone and then we have this brush, right? Mm-hmm. So the brush-like molecules are always moving, right? So they create a thin layer of water at the interface.
Okay.
So that gives you a low coefficient of friction. So we play around with some movement, etc. To be honest with you, even the HA, if you look at it, the brush will give you a better coefficient. In fact, when we introduce the brush, the coefficient of friction is much lower. In this case, what I showed you the data, the coefficient of friction is mostly driven by just the molecular weight, right? Now it is a high molecular weight HA because of the conjugations, right? So that is more like a viscoelastic, right? So the rheological properties are very different.
I see.
So that itself will give you. So 200K is more like a watery, but the minute you introduce hydrogen bonding, you can see the G double prime going up. So G prime going up with the, and so they are going from a viscous behavior to viscoelastic. That itself will give you some low coefficient of friction because they can stretch, right, when you shear them.
Okay. Thank you.
Yeah. Hi, Neil.
Thank you, Shiny, for a really cool talk. Just following up on that question, you showed some data from, I think, in vivo studies that showed that the one that had the UP or UPI motif on the hyaluronic acid, they had a significantly higher retention over a longer period of time. Do you think that's then a sort of viscoelastic effect? Like they're stickier, right? So do you think they stay in the joints because of like a physical chemical effect, or do you think it's also biological?
Physical chemical. I don't think it is the biological. The reason is that so because they can heal back, we always notice that when we rupture them. So there I used my rheology one time. So when you do the step strain measurements, even if you break the chains, they come back as far as they don't move. So in the animals, we saw that unless you use an enzyme to degrade them, they can self-generate the network back. So I think it is mostly because of that physiological aspect is driving. There might be some biological aspect because the inflammation goes down with the time. So that might also contribute a little bit. But I
Uh-huh.
think the initial one is mainly the self-generation of the network and the physical network maintenance of the physical network.
Okay. I know you didn't have time to show this, but do you know what the relaxation times are of these materials? I mean, both sort of the time scales and also the temperature scales of these materials, do they match what you would need in a biological environment like that to sort of make them more slow flowing and more solid like?
Case of the so the temperature we never done. It's always done at 37 degrees centigrade. So I never done what is at lower temperature. So I don't have an answer
Okay.
for that. So if you take the initial, if you do a frequency sweep,
Mm-hmm. Mm-hmm.
in the initial it is more like a solid once you modify them.
Okay.
Because they form this network, right? So because they form that physical network. Now, when you start shearing them, you see that they take over and they ask the frequency. So when you start shearing them, you see that the G prime drop, like the G prime, at high frequency, the G prime takes over. It became more elastic. But in
Yeah.
the walking frequency space, it is more like not a complete fluid, but it's in between. Yeah.
Okay.
Yeah.
Thank you.
Any other questions? Okay, well, Shiny, thank you again so much for your time today. It was a
No,
pleasure hearing about your work. It's beautiful work.
thank you. Thank you.